Welcome to the Health Feeds Performance podcast, where we believe that whole body nourishment is a requisite for athletic success. Tonight we're going to be talking about metabolism, and we've got Emma Beswick from Life Code GX, and I'm really excited by the test that she has, and she kindly did a test with me last year, and it's intrigued me because I've been acutely aware of the differences in carb and fat metabolism for many years. And as a sort of case example, several years ago when I used to cycle in the city behind me, Cape Town, we'd get up early on a Sunday morning and I would need a big fat bowl of porridge for breakfast. Then I'd be having carby snacks during the ride, plus the sports drinks. And it'd be guys I was riding with who would just have a cup of coffee and go for hours. And I always wondered about that metabolism. And in the years since, I've met many people who I would describe as a diesel engine. They just seem to metabolize fat really well. And they don't need much to get going in the morning, whereas you get other people that are more like a high-performance petrol engine that rev up on and need a lot of carbohydrates to sort of fuel that engine. So that's my perspective, and that's how I teach as an example to my students and my clients. But I want to dive in a bit deeper with Emma now, and we'll get into a wee bit biochemistry, but not in huge detail. We want to sort of cover the basis of what is metabolism and what's the relevance within a sporting context. So Emma, a big welcome and thanks for joining me. Why don't you start by introducing yourself and also telling us where your interest in genetics came from and personalization.
Yeah, thank you, Ian. I think we're going to have quite a fascinating talk because my experience personally is very different from yours. And yeah, I think that blend will come out in the wash, as they say. So a little introduction about me. So I'm a nutritional therapist by training. I did that after a long career in corporate world, where my job was really data and analysis and process and designing how things worked, understanding, unpicking, and then putting data back together and creating what we call information, which is something that actually means something as opposed to just lots of things that don't. And so that's really a trait I feel that I've had lifelong. Before I did all that, I did music. And the best part of my music degree for me, again, was the analytical aspect, like taking a score apart, a piece of music, and understanding all the themes and how everything works together. And so that's a theme that has run throughout my life, both from a personal kind of interest, obsessive perspective, as well as a professional one. In terms of genetics, I got into genetics in a way very, very young. So I had an inspirational teacher when I was 15, biology teacher, who set up a project in the school, which was looking at genetics, and we were looking at fruit flies, which are still an organism that is used to do genetic research on. And we all had these fruit flies with different coloured eyes and different shaped wings, and they bred together and turned out, one or the other or somewhere in between. So that was a really, really good start. I did find it fascinating. I loved it. And, I think we can all look back, or a lot of us can look back on our lives and say there was a brilliant teacher there who inspired us and affected us from that point on. So that was always in my head. And then, we're all a blend, I think, in the nutrition world, at least, of our previous lives and histories. And I've done lots and lots of things, but in my leisure time, I've always been interested in running. I've not necessarily been great at running, but I always wanted to be better. And so I started off as a chubby and short 15-year-old, like my mum, and went to uni, got into running, couldn't do much about the height, kind of stuck with that. But I did become very, very fit, lost weight, became lean, and felt really, really good. Then I did a marathon. That's fast forward quite a lot of years. Hated it and did a time of just over 5 hours and just was really upset about the whole thing. A lot of people would think 5 hours is great, but anyway, studying nutrition, got introduced to nutrigenomics, did 23andMe, which people will have heard of, probably massive US company that have sequenced hundreds of thousands, hundreds of millions actually of people's genomes, and started to try and use my analytical unpicking skills to understand a bit more about me and how I could improve my health and my running, essentially. And along the way, I did another marathon, and this time I got a good for age time, something that I wouldn't even have dreamt of. And part of that was my nutrition training. I was still in training, but part of it was learning about genes and personal nutrition, personalized nutrition, and what was right for me. So I was much more scientific about it that second time around. And that then led me to look out in the professional testing marketplace and to understand what was on offer and what wasn't, and to see a real need for something that was very high quality, professional, and from a perspective of functionally minded nutritional therapists and medical professionals and all the flavours of people who are interested in this stuff. So that's ultimately what became Life Code. We went through various stages, but now we offer this end-to-end testing service where we've been able to select our own SNPs, and those are the small changes in our genome, for those who don't know that. So we've got a very bespoke selection that we have researched and determined are the most clinically relevant, most actionable things to know. So that's a little bit of a potted history, because there are billions of these changes that are potential targets for us to analyse, but then you just wouldn't be able to see the wood for the trees. So the selection in terms of what is useful to know is a really important part of that. And the metabolics panel that you mentioned, Ian, that's the latest test that we have that focuses on energy, which is what we're going to be discussing today.
Thanks, Emma. So you've got to this point in your life, like we all have, with a bit of personal journey and interest. So within the space of genetics, and today we're talking about metabolism, if we assume that metabolism does vary a lot amongst different people, from genetic science so far, what has been shown to be the variance? How much variation are we actually talking about? And what does a practitioner kind of need to be aware of when they're working, let's say, with a sports person and carb and fat fueling strategies?
Yeah. So the difference in people can be very significant, and there are certain variances, genetic differences, that actually can have such a big effect that people might be very ill. So those are rare. So we're going to discard them really for the purpose of today. They would be picked up usually when someone's really young, like days or weeks or months old. And they might be so extreme as to indicate that someone cannot digest fats. So they cannot use fats almost at all. That's a very, very extreme scenario. There's certain amino acids that some people just cannot use that would build up and cause toxicity. But what we're talking about are things that are more subtle, that are really common. So the SNPs that we test are a very definition of a SNP, actually. So it stands for single nucleotide polymorphism, but they occur at a frequency of at least 1%. So 1% of a population have the variant, the alternative code. And so that also means that they're quite well understood, because you are able to research them and get good controls versus versus actual targets. So these types of SNPs, they can still be big, but they're probably not going to be life-threatening or life-changing in a medical sense. They are things that can impact, for example, how you digest a certain nutrient or macro, how that is transported through your system into cells, how it's broken down. All of that whole journey of food is impacted by genes. And so some of the magnitude, I kind of like the magnitude of effect. It's like the spike, the difference, a bit like if we were talking about caffeine, how much of a magnitude of effect can a single SNP have? And it can have a lot or it can have a little. We're obviously interested in the ones where it makes a discernible difference. And we're also interested in pathways, so collections of genes and SNPs that together might make a bigger difference than each of the individual parts. And I think that's where the power of nutrigenomics really comes in. It's when you look at contexts, which, there are different types of contexts. There's the context of a sport, and there's a context of a person, and there's a context of a pathway. But we can look at individual ones, and we can look at chains of genes and how they interact with each other and with the wider environment.
So, in other words, a lot of variation.
Yeah.
And you use my favorite word, context or contextualization. So just because science says this doesn't mean it applies to everyone. So context within sport, let's get a wee bit more sport specific now. What are the different contexts within sports that you would consider would affect the importance of metabolism of the substrates?
Yeah. So in sport, you are putting more demand on your system, usually. That's the whole point. And depending how serious you are about that sport, you might want to actually be the best and win. And I think that some of your audience that will apply to, or it will apply to their clients for sure. And so knowing that a SNP can make a big difference or a small difference, even the small difference could be the difference between winning and not winning, because you are fine-tuning your system so much and putting so much demand on it. And there might be weak points in your genetics that mean a person who wasn't putting their body under such amounts of pressure would never know and never notice. It wouldn't make any difference to them. Whereas when you are, it would. So actually, I think this science is more relevant to sports people than the population in general. And certain aspects of it, like metabolism, because we could talk about lots of aspects, but if we're thinking about the need to convert energy into fuel being one thing that is central to most sports performances. Not all, again, it depends on the context, doesn't it? If you're a snooker player, it might be more about the steadiness and the calm and the, but there's certainly some muscle memory in there too.
They use beer to fuel themselves, or they used to. I think they've got a little bit more serious now. Okay, so context is important. The thought that came to me when you were speaking there is Dave Brailsford's famous discussion of marginal gains. So he was the Team Sky and British cycling coach, very famous in that arena. And he talked about marginal gains, which might be aerodynamics or a little bit better quality sleep.
Yeah.
But yeah, adjusting your strategies according to your unique personal needs, you'd probably consider more than a marginal gain in many cases.
Yeah, absolutely.
Should we go a little bit more into the genes on your test and discuss what genes you're looking at and why they're relevant to talk about in a sporting context?
Yeah, absolutely. So the metabolics test, metabolics is a very broad term, isn't it? It could almost mean anything being metabolized and changed. But we wanted to look at energy in, energy out, or fuel in, fuel out, balance of energy, what different individuals do in terms of their biochemistry in directing resources to different places. And a big concept here, I think, is there is this fuel burning aspect, the creation of ATP, but then there is also a kind of opposite to that, which is the restore and the grow. So I'll just share a little visual for those who are watching the visual version of this, but we'll describe it anyway for those who aren't. So we have this concept of when the body, or when our system is low in energy, a whole set of processes should kick in to redress that balance. And that is facilitated by a pathway that we're calling AMPK. So it's basically about there is not enough cellular energy, or it's the battery's kind of gone down, it's depleted. And so we want to be able to convert resources, and that could be fats, it could be sugars, it could be proteins, into ATP ultimately. And that involves a whole chain of events. It's not just one thing. It's actually thousands or tens of thousands of genes are involved in that process. But we know that there are some pivotal ones. But before we get into those, the other side of the equation is a pathway called mTOR. So this stands for mechanistic target of rapamycin. Some people might have heard of rapamycin, especially in the kind of longevity world or discussions that are going on in that space. But really, the important thing about this is it's the opposite of the catabolic AMPK. It's about anabolism. It's about growing, repairing, and this kicks in when there is abundance of resource. So we're fed, for example, we've eaten, and there are sugars or fats or both in circulation, or proteins as well, that are available to our bodies, our biology, and depending on our genes and depending on the pull as well. So the environmental factors are massive here, working together with the genetics. Together that will determine what happens to those resources and where they're directed. So, for example, it could be repairing damage, it could be building muscle, it could be turnover of bone. So both of these aspects of our physiology are important. We need to be able to do both of them, not at the same time because they don't like working at the same time, but they're both really important. And in longevity circles, there is a push to actually try to inhibit mTOR, and that's where this rapamycin element comes in. So effectively, there is a compound called rapamycin that is an inhibitor of this whole growth pathway. And the thinking is that that will help to prevent excess growth, the kind of growth that we might associate with weight gain, the kind of growth that we might associate with things like cancer as well. So it's all got a bit of a bad rap, but actually in the context of sport, we want to be able to rebuild, we want to be able to recover, we want to be strong. And mTOR is really, really, really important for those aspects to be robust and supported. So actually what the target is both. As I said, not at the same time, but in a kind of oscillating way. In a broad way, we could think that we're mostly active in the day as humans, and that's where AMPK, or when AMPK happens most. And at night, it's the mTOR when we're resting, and that repair can happen. So that's some context.
Thanks, Emma. That's one of the best explanations of AMPK and mTOR I've heard, because as you say in the literature, mTOR tends to be the bad guy and AMPK is the good one. But we can't just be catabolic all the time. So that oscillation that you describe, I think, is really, really key. It's the yin yang that I discuss with my clients all the time. Now let's get personal. I see my name on that slide. So presumably the greens and the yellows and the reds are my data. Do you want to go in and share what you found, what really stood out for you when you looked at my test results?
Yeah, I really, really was delighted to see your test results, Ian, because they were a little bit unusual. So that always excites me. It's kind of like, oh, this is interesting. I don't see this very often. So on the visual, for those who have it, under the AMPK pathway, there are two genes that I'm going to drill into a bit and we're going to have a conversation about. One is called PGC1A and the other is called PPARA. Now, these genes are both part of the same family. So genes often come in families of related proteins are made from them. So these genes act in tandem, but they've both got independent, quite important functions. So the first one, PGC1A, this is sometimes referred to as the metabolic master switch. We've got these colloquial terms that sometimes help us remember. But this does control metabolism. It directs whether we are burning, particularly fats, actually. So this is particularly fats. There's a little bit of burning in general, but it's this turning resources into ATP. It stimulates, it kicks off the whole thing. It's kind of at the top of the pyramid, really, top of the chain. And Ian has what we call a yellow dot on that and a little down arrow for those who've got the visual. And what that means is he has a variance. It's heterozygous for those who are into their genetics, which means he's inherited a wild type, which is the more functional version of the gene from one parent, and the potentially less functional allele or letter from another parent. And so you've got that mix, which is heterozygous. And the implication of this, and there are absolutely tons of research which is very consistent about these implications, is that means that gene naturally by default is a little less active, less functional, still functions. So this is the beauty of the genes that we look at. The vast majority of them, we're talking about variances that alter the function by default. They don't block it or stop it entirely. It's like a sliding scale. So your default position is that this is less active in, which for a sports person would seem a little bit odd, wouldn't it? It's like, oh, that didn't sound great. But the beauty is that we can manipulate our genes because the code is an important part of it, but our environment is the other part of the equation. And to add to the manipulable kind of tone, these genes are receptors, or they code for receptors. And receptors in our biochemistry are things that enable us to adapt to our environment. So they sense a certain chemical or chemicals or compounds, and that chemical interacts with the genes, a bit like a baseball catcher, bind to it, and then that sets off a whole chain of events or chains of events. But receptors might downregulate or upregulate depending on how much or how little of these compounds are in the environment. And it's supposed to be a protective mechanism. So, think about caffeine. You drink more caffeine, actually you become more tolerant, and that's your system kind of going, I don't need to be quite so sensitive to this. So I'm downregulating my receptors so that it's not quite so jarring. And we have this ability to adapt, sometimes very quickly. So just because your starting point is down the scale doesn't mean you can't manipulate that and push it up the scale if that's what you want to do.
So just a question on that. I was a middle distance runner and I wasn't great. The longer I went, the worse I got. So could that relate into this pathway a little bit? Because obviously substrate availability becomes more important later on.
Absolutely. So the fact that the next gene down, which is stimulated by PGC1A, so the next gene down, PPAR alpha, is considered to be the biggest single genetic influencer on fat metabolism. And Ian has a homozygous result for that, which is red on our chart, because that's associated with really even further down the scale for the PPAR alpha. And what this is responsible for, it responds to fat actually. So when it senses fats, responds to other things too, but when it senses fats, it kicks into action and it promotes fat uptake, the transport of fats into cells, the breakdown of that fat to acetyl-CoA, the transport into the mitochondria, and the whole of what happens to fat is regulated to some extent by this protein, this receptor that is coded for by the gene. So naturally, your genetic default is that you don't have that sensitivity to fat, which then the sensitivity to it, the sensing of it, then generates all these processes that are about basically metabolizing the fat to use as energy. And your default is like, meh, don't really notice that quite so easily. What was that again? Some fat in the room. Just really not that responsive. And another thing that's really interesting about this is it's quite an unusual, I said that at the beginning, but the PPAR, only 4% of people have, 4 to 5% have a heterozygous result, which is one of the alternate ones, and you've got both the alternate ones. So you're in about 1% of people, maybe less, to have that genotype. So that would, for me, indicate quite strongly that by default, if you're starting off with a clean slate and an even kind of start point, then fats wouldn't be the best fuel for you. But as we said, your body can learn. So it's not that you can't process any fats. It's not that you can't metabolize any fats or utilize them. It just might take a little bit more encouragement and training to start to do that more efficiently. And it might be that even with that, fats are never going to be your preferred fuel.
So is that when I was into several hours of cycling, I needed the regular inputs, perhaps more than other people?
Definitely. So some people might find that, and this is me actually, and we'll come on to my genes in a little bit, but actually it takes me longer to get going, but it's almost like, say I run 6 kilometers, that's when it's almost as if the gear changes and I literally slip into the zone. And it's as if the fat burning has started to give me that fuel supply. And it's the opposite with you. So you deplete your glycogen, and at the time when your body starts to go, what else is there here, please? It's like, no, there isn't really. Not that obviously again. And for you then, what are your options? We could talk about training, but in the short term, your options then are to take in fuel on the go, and the kind of fuels that that are your preference in terms of your biochemistry and your ability to make the most of them. So, but I think the adaptation is an interesting aspect too.
So just a query about that. I'm one of these physiologies that I've always struggled to put weight on, fat or muscle. Do any of these genes have anything to do with that, or are we talking about another sort of range of genetics there?
There are some genes that are a little bit involved in that. So here's all the slides that I'm probably not going to talk about much. There is a gene called AMPD1, and I have it. Here it is. So AMPD1 is actually a gene that recycles it breaks down AMP, which is a signaler to that AMPK pathway. And you've actually got a version of that gene that means your body kind of always thinks that you don't recycle that. You basically your body always thinks, I actually want more fuel. I haven't got enough. Because it genuinely hasn't. So you're losing out the fuel more quickly, if that makes sense. There's a little bit of leakage as well in terms of your available fuel. And that's because, although it's yellow, some people have got a version of this which is down-regulated, which means they are actually benefiting more from the fuel that is in circulation. So there are, I guess, on the fat side of things, there are some things involved in fat storage as well, that you are not someone who tends to store fat safely. So I think this is quite an interesting one. Again, there is a bias against fat in the literature, in all the research. You try and look at studies that talk about high fat diets and also impacts on weight gain, and they're just all a lot of them are very, very biased against too much fat having an effect on BMI. We all know, or most people on this call know, that BMI isn't a good measure of metabolic status or health, because what is that person's body composition? How much of it is muscle and bone and structure? How much of it is fat and how much of it is water, for example? How much lean mass is there? But one thing about fat storage is there is good fat storage and not so good fat storage. And this gene here, for people who don't have the visual, there is a gene called PLIN1. And PLIN1 actually helps people to store fat peripherally. So if you think hips, subcutaneous fat storage, which is considered a safe place to put fat and to put away fat. PLIN1 actually keeps that in a nice, tidy, safe storage for when you might need it. But some people, like you, Ian, have a PLIN1 gene that is less efficient at keeping that subcutaneous, that peripheral fat under wraps. So literally you will leak it out of storage, not in a nice controlled, I need the fat now to use for fuel, but you're just not very good at keeping it in that storeroom. it's as if someone's kind of opened the door and whoever's got in, the bears have got in and stolen all their kind of stores, but it's not there for you. So I think that's quite an interesting one about actually weight and how people deposit fat and where they deposit fat. In terms of sport, that is one potential consideration, like you were saying about, well, is there even any fat to use? You know, how is it stored? And you could think in a really lean athlete, like people doing, extreme cycling events, how much fat is on those guys anyway, to burn? Or, is it really about fueling on the go and being able to get food in, and it's not going to store, it's basically in and through and not diverted anywhere else on the way?
I think that's a really interesting point, because we tend to think for metabolism in an exercise context, it's fat's the slower fuel, carbs the faster fuel, but all of us have enough fat to pull from to provide that base. But you're absolutely right. I mean, some of the elite cyclists have got hardly anything on them. So metabolically, they might be more akin to, going for the easy carbs. But performance-wise, they're very reliant on the carbs quite a lot anyway, because just the intensity they actually get to. So do you want to say just before, I want to sort of take the attention off me, put it over to you in a sec. But before we do that, do you want to just comment on that substrate utilization relevant to intensity? Because I think it's important with the sports context.
Yeah, so I mean, I'm not an expert on the sports context as much as you are, so please do step in. But I think there is an assumption that carbs are more easily utilized. Sugar is more easily utilized than fats. We could talk about ketones, for example. That's another thing. So, what is fat burning? The body doesn't like to burn fats and glucose or sugars or carbs at the same time. So again, there's this alternation between them, which really occurs at the point where these fuels get into mitochondria. And if there is glucose available, so the rapid glucose, the body will always, or most people, and there are some genes involved that might alter this a little bit that I can show you for me. But as a general principle, glucose will be utilized first, and that actually shuts a gate to fat entering mitochondria. So it's as if it's kind of going, no, priority lane is this, and you guys are just going to have to hold back there and wait your turn. And so glucose, that we're showing it down at the bottom of a kind of pathway which shows this entry into mitochondria. Glucose is actually an inhibitor to the fat starting its metabolic process in terms of the actual oxidization and use in the mitochondria to make ATP. So when we've exhausted that carb, that's when that gate opens and lets the fats in. But we still need a little bit of sugar, a tiny bit of sugar, to keep that Krebs cycle going. And the ketone side of things is more extreme, like maybe extreme is the wrong term, but it is kind of a more, right, there is a low carb situation here, there is low availability of sugar, we're going to create ketones. So the ketones are created in the first place in the mitochondria, in the liver, and then burned by the mitochondria in the muscle on the whole. So ketones actually have to go, they have to travel. There's a little bit of in place, but they have to travel. But yeah, I think that I mean, I don't know, Ian, what your experience is in terms of the comparison between carbs as fuel and fats as fuel in terms of timing of availability.
Timing of availability as in the duration of the event, or
as in, like, if you were to consume in event, if you were to drink a blend that had loads of glucose and fructose in it, how quickly can you benefit from that as fuel versus glooping something that is much more fatty?
I've never read that exact information. I just know that the glycolysis pathway is much more rapid than beta oxidation for fat metabolism. So as you say, the carb is always going to be the priority lane. In exercise physiology, we learn that the fat, there's always fat metabolism going on, but as the intensity increases, the fat's the kind of the base metabolism. When we're sitting here now, it's going to be mostly fat, even for me, I'm sure. But then as the intensity increases, then the carbohydrate inclusion becomes more and more. And at the top end of intensity, this is old kind of research, so it might have evolved a wee bit, but the fat metabolism will tend to drop right at the higher anaerobic intensities.
Yeah, and that makes sense to me. You know, logically, it makes sense that is the order of things.
Shall we shift the focus to you for a few moments? Do you want to share your fat metabolism and just sort of comment how it's different from mine and how it would affect how you feel yourself?
Yeah, mine's kind of opposite. But my PGC1A and PPAR alpha are both green. So my whole AMPK pathway, genetically, in terms of the genes and the variances that we're looking at, is as good as it gets, which sounds incredible. But I would say that reflects my actual experience. That, you know, I'm slow on the off, but I hit my zone. And that I actually also like fats more than sugars. So, you know, we're just touching the surface here in terms of, well, there are some genes in some pathways that indicate one person might be more efficient at fat metabolism than another. But that doesn't just impact that particular zoned in small part of your biochemistry. There are all kinds of messaging and sensors going on beyond that, even to the extent of your own intuition and your own food choices, I think. So while my fat burning or fat metabolism genes look pretty good, I actually have a few sticking points on my sugar. So the sugar metabolism pathway that we look at, for example, has some genes called GLUT genes that are responsible for transporting sugars from our gut into our bloodstream and so on and so forth, the journey through our systems. And mine I've got some reds on there, which means they're my nemesis in terms of down the spectrum or down the scale. And I have a bit of a sticky gene called GCK, which is a glucose kinase gene that is actually responsible for glycolysis. So that's right in the middle of the whole how is glucose, specifically glucose, actually broken down and made into acetyl-CoA to then be used for fuel. And one of the important aspects of all of these things is that each individual, when they consume these different macros, it's going from the intestine into our bloodstream, then into the target cells, then into mitochondria, maybe in different forms as it works its way through. But are the places that it's getting stuck. So, for example, for the most part, having lots of fat in your bloodstream or lots of sugar in your bloodstream isn't generally a good thing. It should be temporarily in circulation as part of a delivery mechanism to its destination. So it's part of the journey, but things shouldn't be overly building up in your bloodstream. So I also have a variant on a gene that means that when I do consume fats, so I'm really more efficient at moving fats into my bloodstream in terms of absorption, and that could crowd out glucose. So it's on the one hand a benefit for fat, but a detriment to sugar. And that's actually, for me, intuitively kind of been a thing. So I remember as a kid getting my lunch sandwiches mixed up with my brother's, and he had Nutella on his, and mine were cheese or something, and I actually went without lunch rather than eat these Nutella sandwiches, which sounds a bit kind of extreme. But I think also there is this adaptation aspect, which is if you don't eat sweet things and you have certain genetics that are hypersensitive to sugar or in some way mean that sugar isn't going to be efficiently metabolized, then actually that sugar can seem like a really over intense sensation. And that's how I generally felt as a kid with sugar, that it tasted really, really sweet and it was hugely overpowering. And then when I started doing marathons and stuff, I found it really, really difficult to eat energy products because they were really, really sweet and really, really overpowering for the same reason. And I guess one thing I've learned is you can have carbs that aren't overpowering, that aren't sickly sweet, that actually are more like whole foods and aren't enhanced with extra sugars. But I think I'll never be that person who can just glug a really, really, really sweet drink.
I think that's a key point about, is a carb a carb a carb? Because you find in the I was in South Africa when Tim Noakes got into the low carb discussions, and he just basically blanked out, everything from sickly sweet junk sweets to carrots and apples. And it was a step too far for me because these are real food. So should we be taking food groups out? So these fruits and vegetables contain carbohydrates, but fundamentally they're they're a vegetable or a fruit that have a lot of polyphenols and fiber and short-chain fatty acids and so on. So there's many people that have a soapbox, and we're starting to pick up a wee bit discussion in the chat box about low-carb approaches. And I just want to sort of emphasize this point of my sense over the years of working with multiple clients, many in the sports space, a lot in the health space. And some would do really well on what we used to call the NOACS diet, or we've got mention of Jeff Volek as well, who did research on low carb. And it was very low carb, very high fat. But it's not going to be suitable to everyone, is it? I mean, from what you're sharing tonight, Emma, am I fair in assuming that?
I don't think it would be suitable for you on the whole. And I think, no one is just one or the other, actually. We are designed to be able to benefit from whatever resources are available to us. That is how we survive. That is, that's what makes us resilient as a living thing. So, just because your genetics are showing that fat isn't your preferred thing, and doing a very strict keto diet approach probably wouldn't get the best performance for you ever, however much you try to adapt to it and train into it, doesn't mean you shouldn't eat fat and have good amounts of healthy fats in your diet. And the same with me in reverse. And for these athletes that you're talking about, the NOACS stuff, for example, those foods, as well as being the carb, the macro, also contain, as you said, loads of polyphenols, lots of vitamins, essential nutrients like B vitamins that we need to burn the fuel as well. The fat or sugar doesn't just go into mitochondria and transform on its own. It needs lots and lots of other cofactors to support it. And so what are those cofactors? B vitamins are massive. There's loads of minerals involved. So where are we getting all these things from? If we're eating whole foods, carbohydrate foods are a good source of a lot of those essential nutrients. So it's about balance as always, but perhaps that balance being a little bit different depending on the person and depending on what their goals are and their context.
Thank you. That's really well said. And it's a really important reminder of the cofactors involved in glycolysis, in the Krebs cycle, in oxidative phosphorylation, in beta oxidation, and all the other metabolic pathways in the body and functions. So within sports nutrition, and this is, I got very disillusioned in the 90s when I was studying it because it was white and beige food. And the assumption was that if you get that amount of carbohydrate in your body, you're going to convert that amount to ATP. But what's very clear from your diagram, from your genes here, Emma, is, well, how well genetically are you as a person likely to convert that? And then secondly, are you getting enough cofactors from your diet to support that pathway in the first place? So really, really key considerations. Before we wrap up, I've got one question I'd like to ask you, and then we'll open to questions. Metabolic flexibility. So we've had 50 years of carb is king within sports nutrition. The last 10, 20 years, we've started flirting with fat. Both are quite extreme approaches. one could argue that if you go low carb, high fat, yes, you improve fat adaptability, but the carb burning capabilities might be diminished, and that's when you need to push up a hill or surge for the finish, or have a higher intensity effort. So what are your thoughts on metabolic flexibility?
I think that's the goal for everybody, actually. It's more important the more demands that you're placing upon yourself, but it's how we are designed, how we've evolved. And like you said, you might have a preference for one or the other, but you want to really be able to make the most of these different macros at different times. And you can train to that. So, I think for me personally, knowing what I didn't know when I last did my marathon, I don't know if I'll ever do another one actually, but I can still build in new knowledge. And I think the biggest takeaway from talking to you and discussing this actually is that I would probably benefit from more carbs during an event, because of that. Well, I'm pretty good at the fat usage. Yeah, that's fine. But actually, it's that as the event goes on, or as you want to push harder, maybe you want to save something for the final sprint. Then actually feeding the carbs in to support that during an event, I think is incredibly important. But also making sure that you're trained for that as well, so that you've practiced it and your body's practiced it and it's learned it. So it's not just like, oh, I trained for months on a high fat diet and a low carb thing, and then in the couple of weeks before the event, I just decided that I was going to see if I felt better with some carb on the journey. It's about adaptability isn't instant, and it's about building that into a training program. So you could, you can make use of fats. It's not that you can't. It's just that maybe the proportionality is it's not going to be the same proportion as for me, even if we were doing the same event. So, yeah. Metabolic flexibility is that ability to be able to switch between these fuel types and the resource types and do that efficiently. And that is something that you can train yourself into. I really, really do believe.
Thanks, Emma. I've found that really, really interesting. And I think, nutrigenomics is an evolving science, a very rapidly evolving science. So I think we'll be able to go deeper and deeper as the years unfold as well, and hopefully bring this into the sports nutrition space and the sports medicine space. So just to finish off, if anyone wants to get hold of you or follow your work, where do they need to go?
So it's Life Code GX. So either lifecodegx.com is our website, and if you follow any social, it's at lifecodegx. So those are the best things to do. On our training page on our website, we have lots of webinar links, just a very, very wide range of subjects. Some of the things actually relate to some of the aspects that we've talked about today. So those are, like Ian does, free of charge resources for people to find out more and just choose something that sparks your interest and dip into those. You're very welcome to do that.
Thanks, Emma. More rabbit holes to go down, everyone. I really appreciate the generous input of Emma and all my other guests on the Health Feeds Performance podcast. To listen to or watch more of my interviews, please subscribe to our YouTube channel or any of the other main podcast channels. I'm also really keen to hear what you would like to watch or listen to in future episodes. So please contact us via intsportsnutrition.com. Come through our contact page. And while you're online, have a look at the courses that we run as well. So until next time, bye for now.